Okay, so welcome Biology 30 to the Mendelian genetics unit. We're going to focus on gene linkage in this video. So gene linkage is when you have genes that are found on the same chromosome uh, and they're actually linked. So these two genes, if they're found on the same chromosome, they're going to get passed on together. So linked genes are inherited together. Linked genes are not going to separate during meiosis unless something called crossing over occurs. And remember, crossing over is when two genes that are really close together start to sort of swap portions. And that's what gives you this great picture here, right, where you have the blue chromosome, the green chromosome, the closely tied, physically close portions of those chromosomes are actually going to swap information. That's called crossing over. So unless crossing over is occurring, you won't have linked genes ever separate from each other if they're close together. So I'm going to just walk you through an example. It's going to be maybe a little confusing at first, and then we'll make more sense as we go. But imagine you had chromosome number 13. So this is your first copy. And then you have another copy of chromosome 13. So think of this as, your X, right? Like one dangly chromosome, another dangly chromosome. And this is chromosome 13. When separated during meiosis, one gamete is going to get little a, little b, little c, little d. Another gamete is maybe going to get all the big A, big B, big C, big D. And then the question is, well, hey, what happens if you had a crossing over occur between B and C? So big B and big C. Well, then what would happen is if I crossed over between B and C, one gamete would get little a, little b, and then big C, big D. So we basically cut here right in the middle. One chromosome gets little a, little b, and then big C, big D. The other chromosome is going to get big A, big B, and then little c, little d. So we have recombination occurring here, right? Crossing over shuffled the genetics a little bit. Finding the frequency of recombinants indicates the amount of crossing over. So we can determine how much crossing over has occurred based off of how closely linked genes might be. And this is going to bring us to gene mapping. So gene mapping, typically all gene mapping questions are going to follow the same procedure to answer them. And it's built off of a couple premises. So a gene map is a linear map of genes on a chromosome. So you've got one chromosome, and you've got various genes on that chromosome. For example, eye color, body color, and wing type might all be coded on this one chromosome. We figure out the map of a gene, which chromosomes are on which portion of what gene, by using something called the crossover frequency, COF, crossover frequency. And then spaces between genes are what affect the crossover frequency. So the further apart a gene is, the higher the crossover frequency. So frequency is how often something occurs. So if you have a high crossover frequency, you're going to cross over more often. And genes that are closer together have a lower crossover frequency. So they are not going to cross over as often. And so with that being said, it's quite simple. With crossover frequency, genes that are further apart are more likely to cross over. They're more likely to separate. Or in other words, they're less likely to be inherited or passed on together. Genes that are closer together are less likely to cross over. So in other words, they're more likely to be inherited together. And I'll bring this back to relationship examples like I always do. Think of this as a long distance relationship. If you're in a relationship and you and your partner are provinces apart, you don't see each other very often, you're far apart, you're more likely to make new friends and meet new people, and therefore you're more likely to break up. But if you were closer together in space and time, like you lived in the same city, now you see each other all the time. You're building that relationship stronger, you're seeing each other more often, you're less likely to break up. Well, genes in a single chromosome are no different. further apart the genes are on a chromosome, the more likely they are to break up. The closer together two genes are on a chromosome, the less likely they are to break up. Or in other words, the more likely they are to stick together. And so if you want, you can pause this here and take a look at it. But we're simply saying that alleles closer together generally stay together. Alleles that are further apart on a chromosome usually separate. And so crossover frequency can be expressed in something called map units. Map units are just the term we call the units that separate individual genes on a chromosome. 1% crossover is equal to 1 map unit. So it's basically a percentage. So if you're 4 map units apart, you're 4% likely to cross over. You have a 4% crossover frequency. If you're 6 map units apart, you're 6% likely to cross over. So 50% crossing over frequency is equal to 50 map units apart. So again, don't sweat it. Don't make it more complicated. Map units are a unit of measurement for gene mapping. The greater the map unit, or the distance between two genes, the more likely you are to cross over. Or in other words, the more likely you are to break apart. So the rule for chromosome mapping, when you're doing a question that's asking you to gene map, start with the genes that are furthest apart. If you start with the furthest apart genes, these questions become easy. It's a bit of a puzzle, for sure, but they're not too complicated. If you're a visual person, this is where you want to get a pen and paper out and you want to actually draw these. And do it in pencil because you're going to be erasing and trying to figure out based off what the question's given you. So here's an example. Gene A and B are 22 map units apart. B and C are 24, C and D are 28, D and E are 22. All of them together, so A is 96 map units away from E. So gene E and gene A are 96 map units apart. Or I could say that A and C are 22 plus 24. So gene A and gene C are 46 map units apart, right? A and C are 46 map units apart. You just got to add the two distances up. And so I can safely say that gene A and gene E are more likely to cross over or separate during meiosis than chromosomes B and C, or even B and D, or any of those other combinations that are less far apart. The closer together you are, the more likely you are to stay together. The more far apart you are, the more likely you are to split apart. Okay, so houses on Main Street. Good example. Kind of convoluted, but it's simple when we do the example. So Arnold lives 12 doors away from Beth. Carlos lives 11 doors away from Deanna. Beth lives 3 doors away from Carlos. Arnold lives 4 doors away from Deanna, and Arnold lives 15 doors away from Carlos. So we've got these statements. That's all they are. They're statements. Now, these could be people on a street, or they could be genes on a chromosome. It doesn't matter. The distance between them is measured in map units. That's the distance that they are apart. The rule is always start with the furthest apart. So let's do that. In what order are these houses on the street? Where do these houses on the street line up? Which one comes first, going left to right or right to left? Or in other words, where are the genes on a single chromosome? Well, start with your furthest apart. So Arnold is going to be 15 doors down from Carlos. That's my largest gap. Out of all the information I was given, Arnold and Carlos are 15 doors apart. So I'm always going to start with my furthest apart. So Arnold and Carlos are 15 doors apart. Now I can start filling things in. Well, Beth lives 12 doors away from Arnold. Or I could also say, right, Beth lives 3 doors away from Carlos. It doesn't really matter. Either I put Beth here because she's closer to Carlos, she's only three doors down, Or, based off of our first statement, Arnold lives 12 doors away from Beth. I could put Beth here as well because she's going to be 12 doors, roughly, on my line away from Arnold. Doesn't matter. Both statements tell me that Beth is approximately over here, right? 3 doors away from Carlos, or you could read the statement that she's 12 doors away from Arnold. And so I have options, obviously, because some of you astutely might have drawn Beth over to the left, right? You could have said, okay, well, Arnold lives 12 doors away from Beth. Why did I have to draw Beth to the right of Arnold when I could have drawn Beth to the left of Arnold? Well, you'd be correct. Just based off of that single statement, Arnold living 12 doors away from Beth, you can't really know if Beth is to his left or Beth is to his right. But if you continue to read the other statements, you do have a statement that logically says, well, Beth lives 3 doors away from Carlos. If you put Beth to the left of Arnold, she is not 3 doors away from Carlos. So we can't put Beth to the left. We know it makes the most sense to have Beth to the right of Arnold, right over here. Deanna lives 11 doors away from Carlos. So I could put Deanna here, and that's 11 roughly away from Carlos. I could also use this last statement here. Arnold lives 4 doors away from Deanna. So there's Deanna and Arnold over there, right? So there's Deanna. And now I've got everyone situated. I've got Arnold, I've got Deanna, I've got Beth, I've got Carlos, and I know that everyone's certain distances away from each other. Carlos is 11 doors from Deanna. I can figure that out, right? Because if I know that Arnold and Carlos are 15 apart, Beth is 3 away from Carlos, then the space between here and here is 3. Well, I can figure out the distances between all of these guys by simply subtracting or adding the distances between them from the information that was given to me. It's no different for genes. So let's say Morgan, the guy that really liked his fruit flies, was looking at some data on Drosophila fruit flies. He looked at their eye shape. So they either had a bar-shaped eye or a carnation-shaped eye, so big B or big C. And then they either had fused veins, FV, or they had a scalloped wing shape, capital S. These are all different traits, but they're on the same chromosome. So now we're given some crossover frequencies. COFs are represented in percentage. If they were map units, they'd be represented without the percent. But 1 map unit is equal to 1 COF. So by saying 2.5% crossover frequency, I also mean to say that's 2.5 map units apart. So the FV gene is 2.5 map units away from the B gene. The FV gene is 3 map units away from the C gene. B is 5.5 map units from C. FV is 8 map units away from S. And C is 11 map units away from S as well. So I've got some statements. My rule when I'm figuring out where all these chromosomes are located on a single, where all these genes are located on a single chromosome, I have to start with the furthest apart. So let's start with C and S. So start with C and start with S because they're the furthest apart. They're 11 map units away. So I know from C all the way down to S, that's 11 map units. Now all I have to do is start reading those statements and plugging things in. For example, I know that FV should be 8 map units away from S. And I also know that FV is only 3 map units away from C. So it would make sense to put FV here because it's 3 map units away from C, or my other statement says it's 8 map units away from S. So it should be further from S and closer to C on my line. Right? Don't forget, you could totally have tried to draw the FV on the left-hand side of C because you wouldn't know right away if FV was 3 map units from C to the left of it or to the right of it. But of course, when we look at the second statement, FV is 8 map units from S. There's no way FV could be on the left, just like in our previous neighbors example just before this. So it's not going to be on the left there. B is 5.5 map units away from C. So from here to here, C to B is 5.5 map units. FV and C are 3 map units apart. B and C are 5.5 map units apart. F, V, and S should be 8 map units apart, and I can check that. F, V, and S should be 8 map units apart. And so if I know that it's 5.5 from here to here, then I should be able to determine the difference between the distance of FV and of S, which they are telling me is 8 map units total. So by adding and subtracting all these locations, I should be able to determine which is closest to which and which is furthest away from which. Finally, right, if I go through all of these, I can check my answers. So don't just leave it to chance. You can check if the distances are correct, right? Is FV only 8 map units away from S? Well, you got to play some math, right? So if you know that these should be 8 map units apart, subtract or add as needed to determine if that's correct. So constructing a chromosome map using the following crossover frequencies. So here's another example. You've got a couple different genes and their crossover frequencies, or in this case, their map units, because it's not in a percentage, are located here. And so always start with your furthest apart. My furthest apart seem to be Z and W. And so Z and W are 8 map units apart. Start filling things in. Well, Y and Z are only 1 map unit apart. I could put Y to the left of Z, or I could put Y to the right of Z. But I also know that Y and W are only 7 map units apart. So by putting Y to the right, that's the 8 minus the 1 from here. It makes sense that Y and W are only 7 map units apart. If I put the Y to the left of the Z, it would have been 9 map units apart between that Y and that W, and that doesn't make sense. The X is 2 map units away from Y, or 3 map units away from Z. And so at this point, I have plotted everything. We can check our answers. Well, Z and W are 8 map units apart. We know that. Z and Y were only 1 map unit apart. Well, it says that. X and Y should only be 2 map units apart, and that's correct, right? Between Z and X, it's 3 map units. Y is only 1 map unit away from Z, therefore the space between Y and X should be only 2 map units. Y and W should have 7 map units between them, and again, this makes sense. If it's 8 map units between Z and W, then it should only be 1 less than 8 map units away from Y and W, which is 7, which makes sense. Okay, so at this point, hopefully you can practice some of the examples in the book for gene mapping. I will post these to, well, I'll post a material list in Google Classroom to show you where all those questions might be in your notes package regarding page numbers, but please just flip open your books and take a look at what's what, and do the practice questions here. Don't forget to subscribe to this channel so you can get the next video in the list. Thanks, guys.